in this lecture, we will discuss about replication mechanism in eukaryotes. In the previous lecture, lecture number 11, we discussed about the replication mechanism in prokaryotes. Now, let us continue with the next one. What is that? Replication in eukaryotes. Right? So, there are so many differences between prokaryotic DNA and eukaryotic DNA. Now, in prokaryotes, we told the location of replication in prokaryotes, because nucleus is not there, in the cytoplasm only it will happen. And in prokaryotes, there is no cell cycle, whereas in eukaryotes, means protista, fungi, plantae, animae, all of these are animal kingdoms, they are eukaryotes. Now, here, what is the replication? When does it happen? In eukaryotes, replication occurs. When does it occurs? It occurs during cell cycle. One replication per cell cycle. One replication will happen per cell cycle. We can tell one replication per cell cycle. Now, if you see what is cell cycle. Now, in cell cycle, we have different phases, right? In that, 95% is I phase only 5% is M phase or the division phase. So, this is the M phase or the division phase. So, all this is I phase only. Now, this I phase, which is called interphase, is divided into 3 sub-stages. So, this much is G1 phase, then is S phase, and here is G2 phase. So, the I phase, what is this complete thing called? It is called I phase. The I phase is divided into 3 stages called G1 phase, S phase and G2 phase. Then comes the division phase called M phase. M phase is divided into karyokinesis and cytokinesis. So, here DNA will divide. What is this called? Karyokinesis. And the next one is cytokinesis. So, here we can write it is karyokinesis, which is again divided into prophase, metaphase, anaphase and telophase. The next one is cytoplasm division, is called cytokinesis. This is called cytokinesis. So, karyokinesis, cytokinesis are parts of M phase. I phase is divided into G1 phase, S phase and G2 phase. G1 phase is called first gap phase. What is G1 phase called? It is called first gap phase. After mitosis, it takes a gap. Or we can call G1 phase as post-mitotic phase. After mitosis it is happening, we can also call it as post-mitotic phase. Whereas S phase is called as synthetic phase. S phase is called synthetic phase. So, G2 phase is called as second gap phase. This is the G2 phase. It is called as second gap phase, or before mitosis, so we can call it as pre-mitotic phase. It is also called pre-mitotic phase. Now, why is this called a synthetic phase? Means we tell that in eukaryotes, DNA replication occurs in the S phase of cell cycle. Here it is. So, one replication per cell cycle. When does it happen? It happens during the S phase of cell cycle. DNA replication in eukaryotes occurs in the S phase of cell cycle. In the S phase of cell cycle. Now, if it has to replicate the DNA in the S phase, now it needs so many things, because we told replication is not a simple process, it's a complex process. It needs many enzymes to do replication. Like the main enzyme is DNA dependent DNA polymerase, helicases, SSB proteins. All the machinery and enzymes which are required for replication are synthesized in the G1 phase. So, what it will do in the G1 phase? The cell will synthesize. The cell will synthesize all the enzyme machinery. It will synthesize all the enzyme machinery required for replication, which is required for replication in G1 phase. DNA dependent DNA polymerase, helicases, SSB proteins, what and all it requires for replication are synthesized in the G1 phase. Now using all that, it will do replication in the S phase. Now in the G2 phase, what it will do? It will check for any damage or repair. Before going to M phase, before going to division, it will check S and G2 checkpoint will be there, where it will check whether there is any damage for the DNA, whether there is any mismatches, it will rectify. So in the G1 phase, the cell will prepare itself for the replication. In the S phase, it will do the replication, and in the G2 phase, it checks for mismatches. It checks for mismatches and damage. Is there any damage? Now what it will do, if it finds any mismatch, it will rectify it. If it finds any damage, it will repair it. So that happens in the G2 phase, and once if it ensures that everything has happened perfectly, then only it will enter into M phase. Then only it will enter into M phase. So when does cell division or replication happens in eukaryotes? In eukaryotes, it happens in the S phase, which is in the I phase. In G1 phase, it will synthesize the enzymes and all which are required. In S phase, it will do the replication. In the G2 phase, it checks whether it has done perfect replication or not. And again, so what is a location in eukaryotes where will replication occur? Eukaryote has a eukaryon, true nucleus. So where is the DNA? Inside the nucleus. Then where will replication happen? Inside the nucleus. So location of replication is inside the nucleus. It is inside the nucleus. Replication occurs inside the nucleus itself. And then what is the size of the eukaryotic DNA? We told that the bacterial DNA is small, but if you see, the eukaryotic DNA is big. Take our human DNA, we know that our diploid content is 6.6 into 10 power 9 base pairs. So the size of our DNA, human DNA, if you take, so human DNA size is 6.6 into 10 power 9 base pairs. 6.6 into 10 power 9 base pairs means it is not small DNA, it is big. So if you think that if you start at one place, one particular set of enzymes only can do the entire genome replication, which is so big, it can't do. That means eukaryotic DNAs have many ORI sites. Many ORI sites means from here to here one will do, again here another ORI site is there, from here it will start. So simultaneously it should happen because of the big size of the eukaryotic DNA. So replication starts at a site called ORI site. ORI site has a sequence called autonomous replicating sequence. In prokaryotes, we told there is only one ORI site. It is sufficient because the DNA is small. Whereas in eukaryotes, we told the DNA is big. So then the ORI sites will be many. So then how many replicons are formed? How many units of replications are formed? Many replicons are formed. Because of the bigger size, the number of ORI sites will be many. Many ORI sites are present. Because many ORI sites are present, many replicons are formed. Many replicons are formed. So what do you mean by replicon? Unit of replication is called replicon. And then what is the shape of the bacterial DNA? The shape of the bacterial DNA is circular. But whereas the eukaryotic DNA, it is linear. So what is the shape of the DNA? Eukaryotic DNA is linear. If the eukaryotic DNA is linear, when you open it, DNA helicase will come and it opens. When it opens, then you will get a Y-shaped replication fork. In eukaryotes, a Y-shaped replication fork is formed. In bacteria, you told theta model, bidirectional. This side one replication fork, this side one replication fork. But see here, it is only one replication fork is formed in one direction. So this, because of its linear shape, we can tell Y-shaped replication fork is formed. Y-shaped replication fork is formed. Whereas in bacteria, I showed you replication form this side and replication fork is formed that side also. Now, this is an introduction to eukaryotic replication. So, in eukaryotes, replication occurs during the cell cycle, one replication per cell cycle. In which phase of the cell cycle? In I phase, in the subphase called S phase. Now, if it has to replicate in the S phase, it has to manufacture everything, every enzyme and all. in the G1 phase, ATP, GTP, CTP, it will manufacture in the G1 phase. Using all that enzyme machinery, it will do the replication successfully in the S phase, and in the G2 phase, it checks for mismatches and any damages, and it will rectify it before entering into M phase. And then we told location, because it is eukaryote, it has nucleus, replication occurs inside the nucleus. The size of the eukaryotic DNA is big. We know the size, we have taken the size of a human DNA. It's very big. Such a big DNA, if it has to be replicated from this end to that end, so if you want to do it with one ori site, it's not possible. So that is why they have many ori sites, and then many replicons are formed. The shape, if you take it, is a linear DNA, bacterial DNA is circular DNA, eukaryotic DNA is linear DNA. So when you try to open it, a replication fork will be Y-shaped replication fork formed. So if you want to take the picture, screenshot, you can take this. Now, when we have to discuss about the enzymes of eukaryotic replication. So in bacteria, we told main enzyme which helps in replication is DNA polymerase, is of three types called DNA polymerase 1, DNA polymerase 2, DNA polymerase 3 we told. So the complete name of the enzyme is called as DNA dependent DNA polymerase. The complete name of the DNA polymerase enzyme is DNA dependent DNA polymerase, because the daughter strand synthesis depends upon the bases which are present on the template, right? So the DNA polymerase enzyme, the enzyme reads the base sequence. It reads the base sequence on the template DNA, and it adds complementary bases in the daughter DNA, and it adds complementary bases to daughter DNA. Means the principle behind replication is, what is the principle behind replication is base complementarity only. It is base complementarity only. The principle behind DNA replication is base complementarity only. Now, what are the enzymes? So we know first who will come and identify the ori site and who will come and unwind the DNA helicase. Here also in eukaryotes also, the first enzyme which helps us DNA helicase. So we have seen the detailed functions of all the enzymes in the previous lecture. DNA helicase, it unwinds the DNA and it forms a replication fork. It breaks the hydrogen bonds. DNA helicase, what it will do? It will unwind the DNA and forms a replication fork. Y-shaped replication fork will be formed. Since they are complementary, SSB proteins are required to stabilize the single strands. So SSB means single stranded binding proteins. Single stranded binding proteins. What they will do? Single stranded binding proteins will stabilize the single strand so that they don't recoil. Single stranded binding proteins stabilize single strands. Then we told that the DNA helicase, because it's making a helical structure into a linear structure, so the DNA will face tension and torsion. You have to release the DNA from tension and torsion. If not, DNA will break out. So then you need an enzyme called DNA topoisomerase in eukaryotes, gyrase in the case of prokaryotes. So then we require an enzyme called DNA topoisomerase. So what is DNA topoisomerases? One will be there, two will be there. What will these topoisomerases do? They will relieve the DNA from the tension and torsion created by DNA helicase. So they will relieve the DNA from tension and torsion. From the tension and torsion created by helicase. Wherever there is a tension, it breaks it. It breaks the DNA and again it seals it. Again it joins and it makes a normal strand again. That's the function of DNA topoisomerase. Then who will come? Because we told DNA polymerase can do only elongation and termination. It cannot start the process. Then we require DNA primase enzyme. DNA primase will identify the 3' end of the template and it synthesizes a small primer. So here we can tell in eukaryotes there are almost 9 types of polymerases. There are 9 types of polymerases are there in eukaryotes, whereas in mammalian DNA we can find 5 types. In all eukaryotes, if you group together, there are 9 types of polymerases. But in mammalian cells, 5 types of polymerases are there. What are they? DNA pol alpha, beta, gamma, delta and epsilon. What are the 5 types of polymerases? The first one is DNA polymerase alpha is a first type. So DNA polymerase alpha, it's not just one polypeptide chain, it is made up of 4 subunits. How many are there? Made up of 4 subunits, and it is the largest type of all the 5. It is largest in terms of molecular weight when compared to beta, gamma, delta and epsilon. What will the DNA polymerase alpha do? It acts like a primase. It helps in initiation of replication. So alpha, what it will do? It helps in initiation of replication. Made up of 4 subunits, largest in molecular weight and helps in initiation. It helps in initiation of replication. Alpha helps in initiation of replication. Means it can have polymerizing activity. Coming to what is that 3' to 5' exonuclease activity, 5' to 3' exonuclease activity. This alpha doesn't have any exonuclease. 3' to 5' exonuclease activity, no. 5 to 3' exonuclease activity, again no. It has only polymerizing activity. So alpha do not have 5 to 3 exonuclease activity. It doesn't have 5 to 3 exonuclease activity and no 3 to 5 exonuclease activity. So it doesn't have this activity also, it doesn't have this activity also. It will just initiate the process by making a small primer. What is the length of primer in prokaryotes? We told the length of the primer in prokaryotes is 4 to 12 or 4 to thing. Here in eukaryotes, the length of the primer in eukaryotes, the length of the primer is 4 to 16 nucleotides long. The length of the primer is 4 to 16 nucleotides long. The next enzyme will be DNA polymerase beta. DNA polymerase beta. DNA polymerase alpha, you told it is having 4 subunits. DNA polymerase beta has only 1 subunit and there pol 2 helps in repair, right? It helps in damage. Here also it helps in repair. DNA pol 2 is made up of 1 subunit only and it participates in repair. That's it. It participates in repair. And the third enzyme is DNA polymerase gamma. The third one is DNA polymerase gamma. DNA polymerase gamma has 2 subunits and it is involved in organelle DNA replication, mitochondrial DNA replication. DNA pol gamma is made up of 2 subunits, I am telling, and it helps in mitochondrial DNA replication. It helps in mitochondrial DNA replication and gamma has 3' to 5' exonuclease activity also. So it is having 2 subunits. It helps in mitochondrial DNA replication and it is having 3' to 5' exonuclease activity. It has this activity but no 5 to 3 exonuclease activity. It do not have 5' to 3' exonuclease activity. So alpha, children, it acts like a primase. It initiates the process. Beta, it helps in repair. Okay. Gamma, it helps in mitochondrial DNA replication and mitochondrial DNA repair. Then next one is DNA alpha, beta, gamma and delta. So delta is a principal polymerizing enzyme. Delta is a principal polymerizing enzyme. We know that there are 2 strands. leading strand and lagging strand. Lagging strand synthesis is done by delta. So what strand synthesis delta will do? Lagging strand synthesis delta will do. Lagging strand synthesis delta will do, and delta has, it also has like this enzyme 3' to 5' exonuclease activity it has. 5 to 3 exonuclease activity it doesn't have. So delta, which is the principal polymerizing enzyme, it can do lagging strand synthesis. It has proofreading activity 3 to 5 exonuclease activity means proofreading activity. So it do not have 5 to 3 exonuclease activity. It do not have 5 to 3 exonuclease activity. The last one, we told there are 5 enzymes. The last one is DNA pol epsilon. Alpha, beta, gamma, delta, epsilon. Epsilon helps in the synthesis of leading strand. So it helps in synthesis of leading strand. Leading strand with one primer it will do the continuous synthesis. It helps in synthesis of leading strand. It is made up of one subunit. It is made up of one subunit. It helps in synthesis of leading strand. It also has the same thing, 3 to 5 exonuclease activity, no 5 to 3 exonuclease activity. It also has 3 to 5 for all the things we are telling, 3 to 5 exonuclease activity, but no 5 to 3 exonuclease activity. So all these are the enzymes, DNA dependent DNA polymerases in eukaryotes. In prokaryotes it will pol 1, pol 2, pol 3. Pol 1, it helps in what it will help? It will help in removal of the primer. Pol 2 is inactive, it works only in absence of pol 1. Pol 3 we told it is a principal polymerizing enzyme, but in eukaryotes it is of 5 types: alpha, beta, gamma, delta, epsilon in mammalians. In other organisms 9 types they are telling. So alpha, it helps in initiation. Beta, it helps in repair. Gamma helps in mitochondrial DNA replication. Delta helps in